Hello, am I audible? Hello. Can you please let me know if I'm audible and if my slides are visible? Srikant. Can anyone let me know if I'm audible? Ritika, Srikant. Can you guys turn on your mic and please answer? Okay. If I'm not audible or if my slides are not visible, please turn on your mic and let me know, because once I go into presentation mode, I won't be checking the chat box. Okay, so we have waited long enough, so we'll just start the class now. I hope my slides are visible. Yeah. So welcome to Host Pathogen Interactions live session NPTEL course. This is the session for week 5, and I'm your course TA Sumati from University of Hyderabad, and the course instructor for this course is Professor Himanshu Kumar from ISER. So this week the syllabus included Introduction to Innate Immunity, Introduction of Innate Immunity, Physical, Biochemical and Microbiological Barriers, Introduction to Innate Immunity, Skin, Mucosal Surfaces, Microbiological Barrier, Concept of Pattern Recognition Receptors, that is PRRs, and Pattern Recognition Receptors, TLRs and TLR ligand, then PRRs, TLR signaling, and MID88 and TRIF dependent signaling, TLRs and diseases, and TLRs and therapeutics. So this is a session to help you all with your weekly assignments. So there will be few topics which we will be covering related to each of the assignment questions, which will help you easily answer the questions. If you have any doubts regarding the topics, you can just let me know. Okay, so the first topic is lysozyme and its role in pathogen defense. So the structure in lysozyme function. Lysozyme is an enzymatic protein. So basically it's an enzyme that hydrolyzes beta-1,4 glycosidic bonds in the peptidoglycan layer of bacterial cell walls. So this peptidoglycan layer is the major component of the bacterial cell wall. So if there is hydrolysis of this cell wall uh, layer, then the bacteria will be susceptible to external environment, and the cell wall will be permeable and porous and accessible to other toxic materials which will eventually kill the bacteria. So it is a lysozyme is a muramidase enzyme, meaning it specifically targets the bonds between N-acetyl muramic acid, that is NAM, commonly known as NAM, and N-acetyl glucosamine, also called as NAG. So the bond between NAM and NAG is the one which will be broken. So this forms the basic backbone of your peptidoglycan layer. So this action disrupts the bacterial cell wall, leading to osmotic imbalance and eventual lysis, that is bursting of the cell. So the main function of the lysozyme is to break the bonds between NAM and NAG, your glycosidic bonds, leading to eventual lysis of the cell because of permeability, increased permeability, and leading to osmotic imbalance. So the effect on different its effect on different bacteria. So for example, in gram-positive bacteria, these bacteria, they have a thick peptidoglycan layer, which makes them highly susceptible to lysozyme activity, because this is the major mechanism of action of lysozyme, that is to attack the peptidoglycan layer. So in gram-positive bacteria such as Staphylococcus aureus or Streptococcus pneumoniae and Bacillus species, this peptidoglycan layer is the major component of the cell wall. Lysozyme directly weakens their cell wall, leading to bacterial death. Then in gram-negative bacteria, these bacteria have a thin peptidoglycan layer but are protected by an outer membrane composed of lipopolysaccharides. So this is the one of the major differences between gram-positive and gram-negative bacteria, where in gram-negative bacteria you have your thin peptidoglycan layer, which is then surrounded by a lipopolysaccharide layer, which is your LPS. But in gram-positive bacteria, the outer membrane is the cell wall completely, outer layer is your peptidoglycan layer. So in your gram-negative bacteria, lysozyme alone is less effective because it has LPS layer. But its action can be enhanced by other immune components such as complement proteins, antimicrobial peptides, and bile salts, which help disrupt the outer membrane. So first, to attack the peptido or to get access to the peptidoglycan layer, we have to first eliminate the LPS outer membrane. So when complement proteins or antimicrobial peptides or bile salts or any other chemical which can first clear out the LPS is there, then later your lysozyme can attack the second layer, which is your peptidoglycan layer in gram-negative bacteria. But like how it is very effective in gram-positive bacteria, it is not so effective when there is only lysozyme alone. It needs extra components for the clearance of LPS layer. Then what are the sources of this lysozyme in the body? So lysozyme is widely distributed in the body fluids and also in the immune cells, where it provides the first line of defense against the infection. So since it's the first line of defense, you can see it will be present in most of your cavities, your eyes, your mouth, wherever you have mucus or saliva. So tears and saliva, it is present in tears and saliva, and it protects the mucosal surfaces from bacterial colonization. Then in mucus, in the respiratory and gastrointestinal tracts, it prevents the bacterial overgrowth and helps clear infection. It is also present in neutrophils and macrophages. These white blood cells then release the lysozyme as a part of antimicrobial arsenal. So when your immune cells come into contact with your pathogens, which are bacterial in nature, they then release lysozymes to attack the bacteria. It is also present in breast milk, which provides a passive immunity to the infants. So this helps in protecting the infants against bacterial infections as well. So that's why breast milk is very important for immunity. Then, so it is present in tears, saliva, mucus, neutrophils, and macrophages, which release the lysozyme. It is also present in breast milk. So in all these cases, it helps prevent the colonization of bacteria and help prevent the infection. Synergistic action with other immune mechanisms. So lysozyme does not work alone. It functions in conjugation with other immune defenses like lactoferrin defenses and complement system. So in case what is lactoferrin is it sequesters iron, making it unavailable to bacteria, thereby enhancing the lysozyme's effectiveness on the bacteria. The defensins are small antimicrobial peptides that disrupt the bacterial membranes. Then also your complement system, which helps lyse the gram-negative bacteria, making them more susceptible or more vulnerable to the lysozyme enzyme. So lysozyme is present as a first layer of defense, but it alone cannot eliminate the pathogen or bacteria completely. It needs additional immune mechanism. So it works in concordance with the other antimicrobial defenses or in conjugation with other cells. Then clinical and biotechnological importance. So lysozyme is used as an antimicrobial agent in food preservatives, example in cheese and in wine production. It has potential therapeutic applications in treating bacterial infections, particularly in individuals with compromised immune system. So it can also be given as a therapeutic in people with compromised immune system. It is studied for its role in combating antibiotic resistant bacteria. So they can use, there are several research that has been going on to study how this can help in case of antibiotic resistance. Then finally lysozyme is a critical component of the innate immunity, primarily functioning by breaking down the bacterial cell walls to inhibit bacterial growth and promote the cell lysis by bursting. Then its presence in bodily fluids and in immune cells helps provide continuous protection against pathogens, particularly gram-positive bacteria, making it a key player in the early defense mechanism. So this is a simple illustration of your peptidoglycan layer and how the lysozyme cleaves the bonds between these layers. So here you can see your NAM and NAG are alternative and forming your peptidoglycan layer in the cell wall of the bacteria, and the glycosidic bond between them is broken by your lysozyme. So when these bonds are broken, these NAMs and NAGs are separated and then the cell is then exposed. This is how the lysozyme acts so that your pathogen can be eliminated or your bacteria can be destroyed. Okay, so lysozyme basic structure and function we know now. Then how it is more effective against gram positive and less in gram negative, but along with certain other agents like complement proteins, antimicrobial peptides, etc., it can also, in combination with this, it can help eliminate gram negative bacteria. Then the sources are tears, saliva, mucus, and immune cells like neutrophils and macrophages, and it is also present in breast milk. Then it has synergistic activity along with lactoferrin, defensins, and complement system. So then it can also be used as a therapeutic in immune compromised individuals, and also in it is being studied for its role against antibiotic resistant bacteria. Okay, so the next topic is primary function of TLRs, that is the toll-like receptors in the immune system. Toll-like receptors are a family of pattern recognition receptors, PRRs, that play a crucial role in the innate immunity by detecting the pathogen associated molecular patterns. So PAMPs and DAMPs, that is your damage associated molecular patterns. Both of these patterns are detected by your pattern recognition receptors. And this pattern recognition receptors include your TLRs. This is how you should remember it. Then their primary function is to recognize microbial components and initiate an immune response. This leads to the activation of inflammatory pathways like cytokine production, inflammatory, pro-inflammatory, other interleukin production, etc. Then it eventually helps in adaptive immunity. So how they function is first pathogen recognition. So TLRs identify conserved molecular structures found on pathogens but not in host cells. So these are your pathogen associated molecular patterns, that is your PAMPs. So these include your bacterial lipopolysaccharides, that is your LPS layer, and specific PAMs are detected by specific TLRs. So your LPS is mostly detected by your TLR4, and your peptidoglycan layer, for that you have TLR2. For unmethylated CpG DNA, this is also a pathogen associated marker. So in bacteria and viruses, this is detected by TLR9. And then for viral RNA, we have TLR3, 7, and 8. So these are your PAMs and DAMs, and your TLRs are your pattern recognition receptors. So your PAMs and DAMs are nothing but your molecular structures that are conserved in your pathogens. This help identify them as pathogens. And also these should not be present in the host system. Next is signal transduction and immune activation. So once the TLR binds to a PAMP or a DAM, it activates intracellular signaling pathways, either your NF-kappa B, TNF-alpha release, or anything pro-inflammatory. So this leads to activation of NF-kappa B, IRFs, that is your interferon regulatory factors, and your MAP kinase pathways. So this then will promote the production or release of pro-inflammatory cytokines such as TNF-alpha, IL-6, IL-1 beta, etc. Then it also helps in secretion of type 1 interferon, that is interferon alpha beta production. This is for action against the viruses. Then it also helps in activation of dendritic cells. This enhances the antigen presentation to T cells. So first, pattern recognition is done, specific TLRs for specific patterns, and then signal transduction and immune activation is done to release pro-inflammatory cytokines and production of interferons and activation of dendritic cells for antigen presentation. Then it also bridges innate and adaptive immunity. Okay, so TLR activation enhances the function of dendritic cells and macrophages, which then present the antigens to T cells. This leads to activation of your adaptive immunity. Then this also ensures a rapid innate response followed by a specific and long lasting adaptive response. So it does both. Immediate innate response is activated as well as long lasting adaptive response with memory and very specific to particular type of patterns that have been recognized. Then what is the clinical relevance of TLRs? So in case of autoimmune diseases, overactivation of TLRs is linked to diseases like rheumatoid arthritis, lupus, and IBD. So since we have seen here, it helps in activation of pro-inflammatory cytokines, interferons, and activation of dendritic cells. So if there is any uh, dysfunction or dysregulation of this, it will lead to overactivation of these things. And also it should be very specific in recognizing these patterns on pathogens only and not in host cells. In case if it detects some patterns in host cells, then it will lead to autoimmune diseases. Then infectious diseases. TLRs are critical in defending against bacterial, viral, and fungal infections. So, for elimination of fungal infections, TLRs are very vital. Then it is also used in cancer therapy. So, TLR agonists are being used, explored as immune adjuvants in cancer immunotherapy. So TLR agonists are something that activates the TLRs. So TLRs are generally specific TLRs are have specific ligands. So they will give any such agonist. Agonist is something which is similar to any ligand which can activate that TLR. So this will help activate your immune system, and this is used in cancer immunotherapy. Then also in vaccine development, TLR stimulating molecules, again similar to your agonists. So TLR stimulating molecules are used as vaccine adjuvants to enhance the immune response. So the primary function of TLRs is to act as sensors of infection and tissue damage, triggering innate immune responses and shaping adaptive immunity by detecting conserved microbial and damage signals. So you have both your pathogen associated and damage associated, that is your PAMs and DAMs. They will then help the immune system respond swiftly to infections while maintaining immune homeostasis. So this is your TLR function in a gist. So here you can see they are present on your cell surface and also in your intracellular vesicles. So then you have different TLRs present on different cells. Your TLR2 is present on dendritic cells, monocyte, macrophage, B cell, T cell, microglial cells. 4 is present on dendritic, monocyte, macrophage, B cell, neutrophil and mast cells. Then 5 is present on monocyte, macrophage, T cell and keratinocytes. 3 is present on dendrocyte, macrophage, T cell, microglial and NK cells. 7 and 8 are present on PDC, MDC, monocyte, macrophage, B cell. TLR9 is present on PDC, monocyte, macrophages and B cell. So depending on its role in activation of a particular pathway, it is present on different cells. Then the ligands for them are glycolipids, proteolipids and pathogenic cell wall for TLR2. For 4 it is LPS, host polysaccharides and glycoproteins. Then TLR5 is flagellin. Then 3 is dsRNA. 7 and 8 are your ssRNA. And then for 9 it is unmethylated CpG-DNA. So depending on your whether it attacks your bacteria, virus or your other pathogens, it is present on, it will be activated by different ligands. Then downstream effects is majorly all of them have in release of pro-inflammatory cytokines, your TNF alpha, IL-1 beta, IL-6, IL-10 and IL-12. Other than that, specific TLRs also release interferons, then activate your T cells, then also have chemotactic effects. So depending on the type of TLR, type of PAMP that is present or DAMP that is present, the particular TLRs recognize it and activate the downstream immune response. Then next is your IRAK4 polymorphisms affecting TLR signaling. So IRAK is one of the major kinases that is present downstream of your TLRs. IRAK is interleukin 1 receptor associated kinase. It's a kinase protein. So it is a crucial kinase in the TLR and IL-1 receptor signaling pathways, particularly in activation of NF-kappa B and MAP kinase pathways. So you can see here, this is your TLR. Downstream to TLR, you have MAP, you have your MyD88, and then you have your IRAKs. So again, you have isoforms of IRAKs. You have IRAK1, 2, IRAK4. Then all together, they activate different pathways depending on whatever stimulus is there and whatever TLR is getting activated. Then downstream, eventually you have activation of NF-kappa B pathway, which is important, and then you have other pathways also activated with that is your AP1 IRF pathways for cytokine and chemokine release. Then it also activates NLRP3 inflammasome formation. This is also for immune response, and then it inhibits your oxidative metabolism. So it's involved in many other things, But majorly what we have to know is your IRAC is very crucial for your proper signaling of TLRs. So some known polymorphisms. So IRAC4 polymorphisms can influence immune response because it is a major kinase that is involved, and it makes it susceptible to infections and also can trigger autoimmune diseases. So some known polymorphisms of IRAC4 that can affect the TLR signaling. IRAC4 deficiency or loss of function mutations. So polymorphisms in your IRAC4 are 12x, Q9 293x, E402x in that positions, and other truncating mutations. All these causes loss of function mutations. So certain loss of function mutations in IRAC4 which can introduce a premature stop codon. So truncation of the protein occurs when stop codon is prematurely inserted, or a stop codon due to a mutation, uh, a codon in the middle of the sequence becomes a stop codon. So your entire sequence will not be then translated and the protein will be truncated. That is, it will be cut in the middle itself. So you will not have a complete protein sequence and your truncated protein will be non-functional. So specifically Q287X and Q293X stop codons at position 287 and 293 result in complete IRAC4 deficiency. Then effect on TLR signaling. Impaired signaling is seen through TLR 2, 4, 5, 7 and 9. So it affects basically most of your TLRs, and this leads to deficiency in production of pro-inflammatory cytokines because your pathway will not be now continued downstream. Then your pro-inflammatory cytokines like TNF alpha, IL-6, IL-1 beta, all these will not be produced, or all these will not be secreted to trigger an immune response against your pathogen. Increased susceptibility to gram-positive bacteria. So there is more susceptible, the person becomes more susceptible to bacterial infections like Streptococcus pneumoniae, Staphylococcus aureus, etc., especially during childhood. Then IRAC4 also has single nucleotide polymorphisms, that is your SNPs, which affects your TLR signaling. So these are your S196F. So this affects the single nucleotide polymorphism alters the kinase function, affecting NF kappa B activation downstream. So it can cause recurrence of the infection. An infection can be again be easily recurred because your TLRs are involved both in your innate and adaptive for a long lasting immune response, but then this particular SNP can lead to recurrent infections and may impact inflammatory diseases, including sepsis and autoimmune disorders. Then the other SNPs can affect cytokine production of TNF alpha and IL-6, leading to rheumatoid arthritis, systemic lupus, erythematosus. So your SLE and RA and are also very severely involved in sepsis. Then other SNPs may reduce the IRAC4 function by dysregulation of immune system and inflammatory disorders. So they are eventually, whatever it is, it helps in decreasing the efficiency of your immune response, and less efficiently attacks the pathogens and leads to sepsis, that is your increased infection, and then your autoimmune disorders may arise due to this because your entire now TLR signaling has been dysregulated. So this IRAC4 is very important for TLR mediated signaling, and any loss of function mutation or single polymorphisms, everything will affect its downstream signaling. Then IRAC 4, not only this, but IRAK4 also is involved in autoimmune and inflammatory diseases. So, like previously mentioned, in SLE, RA, and IBD, that is your systemic lupus erythematosus, and rheumatoid arthritis and inflammatory bowel disease. So, this is just examples, were not very specific. So, this is specifically for TLR7 and 9. Similar again, TNF alpha is affected, IL-6 is affected, then all your TLR mediated responses are affected. So in case of inflammatory bowel disease, this also modulates the gut microbiota. So your TLR4 response to gut microbiota gets affected. So that is why your uh, bowel or your gut gets very sensitive because of loss of microbiota. So several IRAK4 polymorphisms, including loss of function mutation, SNPs, different SNPs, significantly affect the TLR-mediated immune response. These variations affect infection susceptibility, inflammation, and autoimmune disease risk, making IRAK4 a key player in host immunity and immune dysregulation. So this is basically affecting all your TLR mediated signaling. Next is consequences of antibiotic treatment on commensal bacteria in microbiome. So antibiotic is generally given as a treatment for elimination of bacterial pathogens, so that uh, your bacterial infection is not established, or even if it is established, it is given to clear the bacteria out of the system as a treatment method. But this antibiotic can also have effects on the residential microbiome that we have in our system, that is your beneficial bacteria. So antibiotic treatment can have significant effects on commensal or beneficial bacteria in the microbiome, leading to various short-term and long-term consequences. So basically, overuse of antibiotics indefinitely will lead to loss of your normal microbiota, gut microbiota, leading to various other side effects, because the body's residential microbiome is involved in many pathways and many uh, signalings which are helping your body have a healthy homeostasis. So, first is disruption of microbial diversity, which is dysbiosis. So, the antibiotics reduce bacterial diversity, often killing both pathogenic and beneficial bacteria. Some bacterial species are more susceptible than others, leading to an imbalance where opportunistic pathogens can thrive. So, basically, one of your key functions of your residential microbiome or your beneficial microbiome already present in your body is it helps you fight against pathogen. It helps as a competitor, or it for space or nutrients, or sometimes it helps recognize it as a pathogen. So in whatever way, it helps your body to prevent establishment of a pathogen. So this will then be affected when your beneficial bacteria are eliminated. Your body becomes more susceptible for these opportunistic pathogens. So these opportunistic pathogens, you are generally do not cause a disease, but when they get a particular situation, then they can become pathogenic in nature. Then overgrowth of opportunistic pathogens. So loss of beneficial bacteria can create an ecological niche for harmful bacteria and fungi to overgrow. So since there were already so many other residential bacteria, these pathogens were not particularly affecting, but because of the loss of the beneficial bacteria, this creates an environment or an ecological niche for these kind of opportunistic bacteria to now grow and thrive in that environment. Example, Clostrio, Clostridioides difficile, that is C. difficile infection, CDI, overgrowth of gut due to loss, overgrowth in the gut due to loss of competing beneficial bacteria. So now that these bacteria do not have competition for space or nutrients with normal other beneficial bacteria, so the Clostridioides difficile now will take opportunity and will now start overgrowing. So example are bacteroids and firmicutes, leading to severe diarrhea and colitis. So your colon infection and diarrhea will be the result. Then candida overgrowth. So in your oral and in your vaginal thrush, there is candida. So this will not generally cause a disease, but due to loss of bacterial species uh, that normally keep the fungal population in check, this candida can now start overgrowing, and once it establishes the colonies, then it will start causing diseases and infections in both oral cavity as well as in vagina. Then next is metabolic and immune system alteration. So the beneficial bacteria, especially present in your gut and your colon, are helpful in processing certain kinds of molecules present in the food, which we generally cannot. So reduced short chain fatty acid production. So this, the butyrate, acetate, propionate, etc., are important for gut health, immune regulation, and inflammation control. So this production will be reduced. Then loss of short chain fatty acid producing bacteria can increase gut permeability or leaky gut. Previously also we have discussed about this leaky gut, and this will lead to inflammation. So the gut bacteria are also very important for essential short chain fatty acid production and also processing other molecules. Then immune system dysregulation. The microbiome plays a key role in immune system education and tolerance. That is, it helps recognize certain things, and it helps uh, to regulate your immune system to tolerate few things and be active against certain things. So this distinguishing factor will also be lost. And then reduced microbial diversity can lead to higher susceptibility to infections, autoimmune diseases, and allergies. So if such distinguishing capacity is lost, then autoimmune diseases also can arise. There will be more allergic reactions. The person will be more susceptible to allergies now, and overall, the immunity will be reduced. Then, antibiotic resistance development. This is one of the major issues that people have been dealing with since overuse of antibiotics or use of unprescribed antibiotics or not completion of the antibiotics treatment course. So several factors affect this. So in case of antibiotic resistance, antibiotics select for resistant bacteria, leading to the spread of antibiotic resistant genes. Then uh, so bacteria have this horizontal gene transfer that they can have, that specific characteristics can be transferred via this horizontal gene transfer from one bacteria to other bacteria. So such kind of antibiotic resistance can also be transferred in such way. Then resistant bacteria can transfer ARGs via horizontal gene transfer to both commensal as well as pathogenic bacteria. So this increases the risk of future antibiotic resistant infections. So once this horizontal gene transfer occurs, of the resistant gene to certain antibiotic. Now, if your body has your pathogenic bacteria also, and this gene transfer has occurred to a particular pathogenic bacteria which is known to be susceptible to particular antibiotic, say a pathogen is susceptible to antibiotic A. But due to this horizontal gene transfer, now this pathogenic bacteria has a gene which helps it resist this antibiotic A. So now when this antibiotic A is given as treatment against this particular bacteria, it will not work. It will not help eliminate this bacteria. Now then again, different antibiotic combinations has to be given, or then there have to be screening that this bacteria, they will take the culture and they will screen that what are the antibiotics this particular bacteria is now again resistant to and susceptible to. Based on that, then again combination drugs will be given. So all these will increase due to antibiotic, which will clear out, or excessive use or abuse of antibiotics particularly, leading to all these kinds of issues, mainly affecting your gut microbiome, immune system, helping your opportunistic bacteria to thrive, and leading to antibiotic resistant bacterial strains. So we can see this. We have different types of microbes that are present in our gut. So every microbe has certain particular help or certain particular benefits it gives the host. So it lives in commensalism. So it few of them help for enzymes for biotransformation. Then few of them help in maintaining the mucus layer. Few of them help in cytokine production. Few of them help in fermentation, production of short chain fatty acids. Few of them help in enzymatic activity, then glucose metabolism, energy intake, and many of them help preventing the biofilm formation. Sometimes they can produce even antimicrobial peptides, and it can prevent opportunistic bacteria or pathogen to grow and establish an infection. So all these kinds of benefits are affected due to antibiotics, or it can also lead to antibiotic resistance. Then long-term health consequences. So it increases the risk of chronic diseases. So dysbiosis has been linked to obesity, diabetes, inflammatory bowel disease, allergies, neurological conditions, disrupting the gut-brain axis, then also delayed microbiome recovery. So the gut microbiome can partially recover after antibiotic treatment, but some bacterial populations may never fully return to their original diversity. Repeated antibiotic use can cause cumulative damage to microbiome. So this recovery of gut microbiome is very important. So even during drug testing and clinical trials of a antibiotic, etc., they will also check whether they will check for a particular dose or particular time that it has to be given, so that it can eliminate your pathogenic bacteria, but at the same time it should give chance after the antibiotic treatment is stopped, your gut microbiome should be able to recover and establish your normal commensal microbiome in your system. If this does not occur, then all these problems will arise. Then this is mainly due to, as I said, abuse or overuse or repeated antibiotic use. That is why you should not, take unprescribed antibiotics, or not uh, complete the total antibiotic course. So then what are the mitigation strategies for this? So what can we do to help overcome this? We then have probiotics and prebiotics. So this help restore the beneficial bacteria and support gut health. So your probiotics are basically your probiotic bacteria, and then your prebiotics are something that helps it grow and recover your gut bacteria and support good gut health. Then fecal microbiota transplant. So in extreme cases, this is done where your body has completely lost its microbiota and there is no chance of recovery, and it leads to several such conditions like IBD, obesity, diabetes, etc. Then fecal microbiota transplantation is done. So used for severe C. difficile infections to restore the microbiome balance. So what they do is they extract the microbiota, gut microbiota from feces, they purify it, they then again transfer it into the gut so that they can establish a microbiome environment now. Then narrow spectrum antibiotics reduce broad microbiome disruption by targeting specific pathogens. So the antibiotics, there are specific antibiotics to specific pathogens. Then, like I said, they screen it and then they give which infection we need what kind of antibiotic. And there are broad spectrum antibiotics and narrow spectrum antibiotics. So broad spectrum antibiotics can disrupt many types or many different microbes at the same time, but narrow spectrum is specific to one particular type of pathogens. So if we can identify what pathogen it is specifically, then we can use narrow spectrum antibiotics so that it doesn't affect the other species which are not related to this kind of pathogen. So all these ways we can help uh, recover the commensal microbiome. Then so finally, antibiotics disrupt the commensal bacteria, leading to microbiome imbalance, that is nothing but dysbiosis, loss of beneficial microbes, pathogen overgrowth, metabolic changes, immune system dysregulation, and antibiotic resistance. So, careful use of antibiotics along with microbiome supporting strategies is key and is crucial to minimize these kind of side effects or negative effects. Next is the characteristics of innate immunity that make it essential for survival. So what are this is related to the question we have directly. So what are the key characters that are correct in that are it acts as the first line of defense against microbes. So innate immunity, first line of defense. Innate immunity provides immediate protection against pathogens through physical barriers, skin, mucosa, antibiotic enzymes, antimicrobial enzymes like lysozyme, and immune cells like macrophages and neutrophils. So, innate immunity is your first line of defense because it has all these uh, immediate first line of defense layers, that is your skin, your mucus layer, and then your lysozymes which is present in your mucus, saliva, tears, etc., and then your immune cells which attack your pathogens or infections immediately. First are your neutrophils and your macrophages. Then it initiates an immune response within hours of exposure. So it is immediate response, so rapid. It should be within minutes or within hours. The trigger, the activation of immune system is within hours. So unlike adaptive immunity which takes days to respond, innate immunity detects and responds to the infections rapidly within hours via pattern recognition receptors like TLRs and inflammasomes. So we saw that the TLRs, which are a part of the pattern recognition receptors, and the inflammasome, which we saw here. Yeah. So your TLR also basically uh, activate your inflammasome. So this together help in activating immediate response to the uh, infection. Then it compromises an ancient and evolved system. So innate immunity is evolutionarily conserved. It comprises an ancient and evolved system. It is evolutionarily conserved across species, existing in vertebrates, invertebrates, and even in plants. So basically, it is conserved via different classes of living organism, making it fundamental component of the immune response. So it is ancient, it is evolved, and it is conserved via various degrees or various classes of organisms. Then it recruits effector cells to fight infections. So innate immune mechanism triggers inflammation and recruits immune cells. Example, your neutrophils, macrophages, and NK cells to the site of infection, and it helps to contain and eliminate the pathogen. So, recruiting the effector cells to fight your infection is also an immediate response within hours. So, your innate immunity triggers then your inflammation, that is, your pro inflammatory cytokines and all are released, and then your neutrophils, macrophages, NK cells will be recruited at the site of infection. Then it influences the health through the microbial gene pool. Yeah. So the innate immune system interacts with your microbiome, shaping the immune tolerance, inflammation, disease susceptibility. Beneficial bacteria can influence immune response, and innate immunity regulates microbial composition. So it's both ways. Your in microbiome helps your innate immunity to have specific response to specific pathogens and have tolerance, and sometimes helps stimulate or activate inflammation. And also the innate immunity itself also regulates the microbial composition. So your beneficial or your commensal microbiome is preferred, and your opportunistic microbiome is kept at check. So your beneficial bacteria can influence the immune response, and innate immunity regulates the microbial composition. So what are the correct options for these? That is the characteristics of immunity that makes it essential for survival. Is it acts as the first line of defense against microbes. It initiates an immune response within hours of exposure. It comprises an ancient and evolved system, that is a conserved evolutionary pathway, and it recruits effector cells to the site of infection to fight infections, and it influences the health through the microbial gene pool. Next is the mannose binding lectin. So mannose binding lectin, or MBL, to the it belongs to the collectin family of immune proteins, which are part of the C-type lectin superfamily. The main function of MBL, or mannose binding lectin, is a pattern recognition receptor, and it helps in the innate immunity and activate the complement system via the lectin pathway. So the complement, you have your classical pathway, your lectin pathway, and your other alternative pathway. So your MBL is part of your pattern recognition receptor, PRRs. Like how TLRs are also part of your pattern recognition, your mannose binding lectin is also part of your pattern recognition receptors. And then it helps in activation of complement system via the lectin pathway, contributing to innate immunity. Functions of MBL in immunity. Since it is a pattern recognition receptor, pathogen recognition and binding is done. So MBL recognizes mannose. That is why it is a mannose binding lectin. So it recognizes mannose, fucose, and glucnac. That is your N-acetyl glucosamine on the microbial surfaces like bacteria, fungi, viruses, parasites, etc. So all the pathogens that contain your mannose, fucose, and glucnac on the surfaces, these kind of residues are uh, recognized, and they bind to the mannose binding lectin. Since these sugar patterns are common in pathogens but rare in humans, so these are, since we have to have the PAMPs, these are basically the PAMPs. So these PAMPs are specific to the pathogens and not very and are very rare in humans. So these help in MBL and your activation of your lectin pathway. And MBL acts as a selective detector of foreign invaders. Then next is activation of your, just a second. Yeah. So after pathogen recognition and binding, next is activation of your lectin complement pathway. So because that is downstream of your MBL. So MBL binds to the microbial surfaces and associates with the MBL associated serine proteases, your MASPs. So these MBL associated serine proteases cleave C4 and C2, leading to the formation of C3 convertase in your complement system. So which then enhances opsonization, inflammation, and membrane attack complex formation. So first, your MBL recognizes the PAMs, that is your sugar patterns, your carbohydrate patterns, this glucanac, fucose, mannose, etc. Then once it selectively binds to these, then lectin pathway is activated by uh, MBL associated serine protease, that is your MASP. Then it cleaves the C4 and C2, and C3 convertase formation is done. Then we have opsonization. So opsonization is nothing but it helps the body's immune system to identify and destroy a pathogen or anything that is foreign. So this helps in the opsonization also helps in uh, pathogen destruction, or it the opsonized pathogen can then be again, recognized by your uh, immune cells, like your phagocytic cells like macrophages, neurophages, dendritic cells, etc. And then these phagocytic cells will then eliminate this. So that is your opsonization. And your inflammation, release of your pro-inflammatory cytokines, recruitment of your immune cells, etc. Then we also have membrane attack complex. So then there will be uh, formation of this um, membrane attack complex. So this is also known as MAC, and it basically is a protein complex, and it helps in pore formation in the pathogen membranes. And then your complement system, other proteins like your C6, C5b, C7, etc. will then uh, the, uh, this is part of this MAC formation. And then your osmolysis will occur because of the increased porosity of your pathogen. So MAC can also then trigger your intracellular signaling and cell activation. So this is your opsonization and inflammation and membrane attack complex formation. With your membrane attack complex also consists of several other complement proteins like C5b, C6, C7, etc. So opsonization and phagocytosis. MBL tags pathogen for destruction by binding to the phagocytic receptors on the macrophages and neutrophils, enhancing phagocytosis. So eventually phagocytosis will eliminate or destroy these pathogens. Then neutralization of pathogens and modulation of inflammation. MBL can directly block the viral entry, um, like the influenza, SARS-CoV, etc., by binding to the viral glycoproteins directly. Then it also regulates the inflammatory responses to prevent excessive tissue damage. Clinical relevance of MBL. So MBL deficiency is also there. So in this case, it increases the susceptibility to recurrent infection, similar to your TLR dysfunctioning due to IRAC, because this is also a PRR which detects your PAMs. So there is also again recurrent infections, and especially in infants and in immunocompromised individuals. Just a second. Yeah. So yeah, so it is also associated with autoimmune diseases, example lupus, rheumatoid arthritis, etc. Then therapeutic potential is MBL based therapies are being explored for human deficiencies and sepsis management. Next is biochemical and chemical barriers involved in innate immunity. So here also we have the correct choices or the correct options. So first is lysozyme breaks down peptidoglycan layer in bacterial cell wall, just like we discussed. It is an enzyme found in tears, saliva, mucus, phagocytes, etc. It hydrolyzes beta-1,4 glycosidic bond and peptidoglycan, leading to bacterial cell wall degradation. Most effective in gram-positive. Next, MBL binds to sugars on the pathogen surface, that is your mannose, fucose, and glucanac. So it activates the lectin pathway of the complement, enhancing the opsonization and pathogen clearance. Then alpha defensins are antimicrobial peptides produced by the gut and the lungs. Alpha defensins are small cationic peptides produced by Paneth cells in the gut and epithelial cells in the lungs. They disrupt microbial membranes by forming pores, leading to pathogen lysis. So defensins also we discussed. Uh, Cathelicidin is an antimicrobial peptide stored in lamellar bodies. So cathelicidin's example, your LL37, are antimicrobial peptides stored in lamellar bodies of the keratinocytes. That's why one of the TLRs were also present in the keratinocytes, because keratinocytes also help in immune response in the skin. They play a key role in skin defense by disrupting the microbial membranes and modulating the inflammation. So, cathelicidin is present in the lamellar bodies of the keratinocytes and help modulate the inflammation. Histatins are antimicrobial chemicals present in the oral cavity. So histatins are also cationic antimicrobial peptides, but this is found in the saliva. They are effective against fungi like Candida albicans and some bacteria, protecting the oral mucosa. So all the correct options are lysozyme breaks down peptidoglycan in bacterial cell walls. Mannose binding lectin, that is MBL, binds to sugars on pathogen surface. Alpha defensins are antimicrobial peptides produced by the gut and lungs. Cathelicidin is an antimicrobial peptide stored in lamellar bodies of the keratinocytes. Histatins are antimicrobial chemicals present in the oral cavity. Next is PAMs. PAMs are pathogen associated molecular patterns. So, PAMs are conserved molecular structures found on pathogens but not on host. This we know. So, it is recognized by the innate immune system to trigger an immune response. What are the basic characteristics of PAMs are? It is highly conserved molecular pattern. It is unique to pathogens. It is recognized by the PRRs. It triggers an immune response, and this helps in eliminating or fight the pathogen or infection. So PAMs are essential for pathogen survival, meaning they do not easily mutate to evade immune detection. So why these are conserved molecular patterns are because they help for the pathogen to survive. It is actually essential for the pathogen. Our body is using this essential survival pattern or essential survival uh, biomolecule or anything that is required for the pathogen to survive as a detector to or as a signal to detect or sense that it is a pathogen. So since they are essential for the pathogen to survive, they will not be easily changed or easily mutated. That is the reason they are conserved, and it helps our host system to easily identify this, because there will be no very frequent changes in this, so it will be easy for the immune system to set it as a standard for recognition of a pathogen. Then the immune system distinguishes self from non-self by recognizing the PAMs, preventing autoimmune diseases, because PAMs are only present on the pathogens but not in the host cells. Then PAMs are detected in by innate immune receptors such as Toll-like receptors, Nod-like receptors, RIG1-like receptors, C-type lectin receptors, etc. So this detection of PAMS leads to cytokine release, inflammation, complement activation, and phagocytosis, eventually to eliminate the pathogen. So some examples of PAMS and their associated microbes are LPS is one of the very major known PAM. It is present in gram-negative bacteria and it is recognized by TLR4. And then we have peptidoglycan present in gram-positive bacteria, which recognized by TLR2, NOD1, NOD2 receptors. Then lipoticoic acid, LTA, that is also present in gram-positive bacteria, recognized by TLR2. Then flagellin, which is present in the flagella of bacteria, flagellated bacteria. So this flagellar protein is also recognized as a PAMP by TLR5. Then we have unmethylated CBG islands, which is very common in bacteria and viruses, recognized by TLR9. Then DS RNA, that is double-stranded RNA present in viruses, is recognized by TLR3, RIG1 and MDF5. Single-stranded RNA, ssRNA, is present in RNA viruses and is recognized by TLR7 and TLR8. Then beta-glucans that are present in fungi are detected by detectin-1 and TLR2. Mannan, which is detected by fungi, which is detected by C-type lectins, are present in fungi and some bacteria. So basically, PAMs are conserved molecular patterns found in microbes but not in host cells. They are recognized by PRRs to initiate the immune response. And the examples are whatever we saw here: LPS, flagellin, DSD, RNA, beta glucans, etc. Then pattern recognition receptors are a group of receptors that are that recognize molecular structures on pathogens. So PRRs are the ones which recognize the PAMs. So that is why they are the receptors for the PAMs and they recognize the molecular structures present on the PAMs. Not only PAMs, they also recognize the molecular structures that are prominent or characteristic features of damaged cells and apoptotic host cells. Those are DAMs, damage associated molecular patterns. So they play a key role in innate immune response, that is help in recognition and then activation of specific immune response against it. There are different type of PRRs. We have nucleotide binding oligomerization domain, that is NOD-like receptors, and these help recognize the pathogens and activate the immune response. Then we have RIG1-like receptors. These are intracellular receptors, recognize viral nucleic acid and trigger antiviral immune response. Then we also have cytosolic DNA sensors. These receptors identify the DNA viruses such as cytomegalovirus and vaccinia virus. Then detectin-1 recognizes beta glucans, which are polymers of glucose present in fungal cell wall. Then scavenger receptors. These receptors bind to bacterial cell wall components such as LPS. Then toll-like receptors, they bind to different stimulus as we discussed earlier and include many like 7, 9, 2, 3, etc. And PRRs trigger signaling pathway that activate a defense program called pattern triggered immunity, that is PTI. Then they bridge non-specific and specific immunity. So we can see these are the glycans of the lipopolysaccharide, then we have proteins, then we have zymosan for nucleic acids, and these are all your PAMs and your DAMs, which will be recognized by your innate immune cells or your PRRs present on the cells. So we have the cells, mainly macrophages, dendritic cells, neutrophils, and natural killer cells, and immune cell pattern recognition receptors, that is your PRRs, your toll-like receptors, NOD-like receptors, and C-type lectin. So family of pattern recognition receptors primarily responsible for sensing are your RNA viruses. We have RIG1-like receptor, family primarily for sensing RNA viruses. Then PRR family for RNA virus detection. RIG1-like receptors are cytoplasmic sensors that detect viral RNA inside the infected cell. They recognize viral double-stranded RNA and also uncapped single-stranded RNA. Then activation of RLRs uh, triggers antiviral immune response, including type 1 interferon production. So major RLRs involved in RNA virus detection are RIG1, MDF5, LPG2, RIG1 retinoic acid inducible gene recognizes short double-stranded RNA and 5' triphosphate single-stranded RNA, commonly present in many RNA viruses. It also detects influenza A, rabies virus, hepatitis C virus. Then MDA5 is melanoma dissociation associated protein. This detects long dsRNA found in picornaviruses, example poliovirus, and LGP2 uh, regulates RIG1 MDA5 signaling acting as co-receptor. Then other PRR families involved in RNA virus sensing are toll-like receptors, endosomal receptors. TLR3 detects double-stranded RNA. TLR7 and 8 detects single-stranded RNA. Nod-like receptors are involved in inflammasome activation, and NLRP3, that is inflammasome, uh, senses the RNA virus by detecting cellular stress and danger signals. Next is significance of TIR domain in the toll-like receptors. So toll-like receptors or interleukin 1 receptor have TI, that is the TIR domain is conserved in intracellular signaling domains of TLRs. That is essential for initiating the innate immune response. So we can see here these are your toll-like receptors, and we have your TIR present in your toll-like receptors, which help in binding with your adapter proteins. So signal transduction in innate immunity. The TIR is responsible for intracellular signaling after TLR detects a pathogen-associated molecular pattern. It mediates the interaction with adapter proteins that can activate the downstream immune response. Then recruitment of the adapter proteins. The TIR domain enables the TLRs to recruit the adapter proteins such as MYD88, TRIF, TIRAP, MAL, TRAM, etc. So these activate MYD88 activates NF-kappa B and inflammatory cytokines. TRIF activates interferon response. TIRAP and MAL activates or helps in MYD88 to certain TLRs. Then TRAM links TRIF to TLR signaling. So all these are connected downstream signaling. You can see here MYD88, NF-kappa B, MAL, NF-kappa B. TRIF is involved in NF-kappa B and interfer IRF3, IRF5 and IRF7. And then TRAM is involved in NF-kappa B, IRF, and it also is regulated here. And then we also have other signalings which are in MDC cells. So that is it for this assignment. If there is any doubt, you can ask me or you can mail me to this email ID. Your queries will be addressed.